Hello, we are at number 6 and we will explain our STEM project. The name of this is Cellergy. The main subject of this is the water pollution caused by organic waste and the lack of clean energy source. The SDG involves are SDG 6, the clean water and sanitation, the SDG 7, the affordable and clean energy, and SDG 13, the climate action. The key characteristics are the water pollute the environment, while the demand for this keeps rising. As a sustainable solution, must boost and reduce the environment contamination and the generate clean energy. The challenge for this is the water pollution and the energy demand. For air pollution and fossil fuels or water use degradation and the climate. Our action is need for natural resource protection and environmental justice.
Mathematical knowledge, it has basic math, radius, voltage, and formulas to electricity measure. Then it's why this project is to explore how can turn waste into electricity. And the next is the goal, and is microbial fuel cell. A microbial fuel cell produces electricity from bacteria and understand how it works. And then its importance is to provide eco-friendly energy and reduces the pollution. And benefits is clean energy, and waste reduction and hands-on learning about how science applies to real life.
Now the proposed solution. Here we propose building a microbial fuel cell using compost as water contamination too, as the bacterial source. We have the environmental benefits. It reduces soil pollution while generating clean energy. Also helps decrease the use of fossil fuels. The viability requires simple materials, as you can see here, 2 chambers, electrodes, and a proton exchange membrane. Natural bacterial communities in compost already work for this purpose. It is inexpensive and possible to test in a small scale. And the product purpose: to show that organic waste can generate electricity, to measure which bacterial sources produce the best results, and also to demonstrate a practical and eco-friendly innovation.
CDJ 6, clean water and sanitation, reduces the water pollution with compost. CDJ 7, the microbial fuel cell, produce electricity with compost. And CDJ 13, by reducing reliance on fossil fuels, and Cellergy helps lower greenhouse emissions.
The theoretical framework. The growing and even better waste management creates a major using of this project. We have that the fossil fuels create pollutions and climate change, while also the unburied organic waste contaminates makes soil pollutions and contaminates water and harms the ecosystem. So our project attacks these both problems: the waste of organic waste of organic waste, and the fossil fuels pollution of the soil pollution in the ecosystems. We have that our microbial fuel cell converts this cell energy converts the organic waste into electricity using bacteria. And we have the exoelectrogenesis, that are the bacteria shown in our project. This transfers electron to the anode. And we have the redox reaction, that is the reaction that makes our project inside of it. That is the bacteria break down the waste of the organic waste, produce electrons and protons, and this is the electricity that it creates. We also have the scientific validation for our project. We have these few points that studies show that these MFC, that is microbial fuel cells, can sustainably generate electricity from the bacteria that use the decomposition of the waste. We have efficiency depends on the exoelectrogenic bacteria, that is what we see in the slide before. And we have also these few points that are the carbon-based materials improve bacterial attachment. The oxygen-rich cathode is needed to complete the circuit that we have. We need the cathode that get it's a dependent variable, because it gets into the prototype and then it goes out like an anode to create the electricity. And we have these findings, these scientific findings, are used to our validation. This proves that our Cellergy prototype truly works with these bacteria to generate electricity. For our chemical phenomena, we have three important points to prove how this organic waste gets into the project and produce electricity. We have the first point is the anode reaction, that is the oxidation in another terms, that is inside of the chamber, inside of the prototype, we get the bacteria. Like, for example, there are two examples of bacteria, the Geobacter and the Salmonella. They are the metabolized organic compounds, and during the process they release the anode. They are like the cathode, and they release the anode. We have the electron flow. Inside of the chamber, the electrons flow in an external circuit to produce the electricity, the electron current, and it can be measured with a multimeter. And finally, we have the cathode reaction, that is the final step that it creates energy. We have at the cathode, the oxygen reacts with all the incoming electrons and protons that flow into the current of the prototype, and it forms water. Then with that water, it produces a redox reaction that it completes the full circuit. And when we complete the full circuit, it creates the electron flow, and that electron flow is electricity. And like a little summary, we have these are steps from how it works. We have first the location of process key, that is the reaction. Then we have the first thing of the prototype, the anode reaction, the oxidation of the bacteria, the organic matter that is produced. Then we have the external circuit, that is the current flow of the electrons. We have the electron transfers, and then the electrons that are flowing into the cathode, that I told you that it was the last step. And we have the cathode chamber, we produce the redox reaction, and the oxygen that it converts into water to produce it.
Well, we have the prototype blueprint. I made the prototype of our STEM, and I see that, we have two cylinders. The first one have composed from Starbucks coffee, and the second one have oxygenated water. This is a salt bridge containing both chambers. These have grenadine and salt, and its mix make the salt bridge. we have two electrodes that are connected by two pieces and are connected to the multimeter that shows a voltage of 0.22. So, but if you only connect one electrode, I don't know, the water oxygenated or the compound no makes any voltage. So only if you connect twice, you make a voltage. We have a better description about the project. Well, we have another chamber that is the first cylindrical with compost and with the bacteria. Second, we have the second cylinder with oxygenated water. Then we have the tube with resin and water. Then we have the graphite root. The graphite are the electrodes that is into the cylinders. Then we have the copper wire connected to 12. Then we have a platform made of wood that I put the two cylinders. In resin, decomposing another chamber provides electronic bacteria. Electrons flow from the anode to the cathode through the external circuit. Product travels through the salt bridge to complete the reaction.
The expected function. Our breed use serves to sustain MFC. They use a continuous Redox cycle to convert organic waste into electricity. In the anode chamber, the bacteria consume organic matter, releasing electrons, protons, and CO2 and CO2. And then the electrons transfer to anode, then to electric circuit, and then to electric current. The protons migrate through proton exchange membrane to cathode. The cathode chamber is when electrons and protons produce H2O. Well, the variable table, the voltage generated is the electrical potential difference between electrodes. We use a multimeter to measure, and the specific results are 0.3 to 0.6 volts. The electric current is when flow of electrons through the circuit. We also use a multimeter, and the spectral results are 1 to 5. The compost pH is the acid level of the compost mixture. We use a digital pH meter, and the spectral results are 6 to 7. The temperature is the internal temperature of the system. We use a thermometer. The spectral results are 20 to 30 centimeters. And generating time is the time until voltage is detected. We use a stopwatch. The spectral results are 10 to 20 minutes. The measurement plans. The objective is to validate the electrical performance and stability of the MFCs. The measurements are the voltage and current. We recorded like until 7 days to see what can change in their internal condition. The power regulation is the power and voltage per current. Our goal is confirm the MFC efficiently converts chemical energy into electricity and aces its potential as a stable, eco-friendly, and expected benefits. We expect to operate to generate clean energy, to convert the chemical energy from the compost into electricity via bacteria, cleaner than fossil fuels, and provides a renewable power source. Also to repurpose organic waste, uses compost as fuel, reducing landfill volume and methane emissions, turns waste into a useful resource. Also to learn real-world biology and chemistry, how are they related as science, how biology studies the microbial metabolism and exoelectrogenic bacteria, and chemistry understand the electrochemical principles. And to support these global sustainability goals, as you can see here, such as affordable and clean energy, sustainable cities and communities, and responsible consumption and production.
The compost-based microbial fuel cell is more than a science project. It connects sustainability with education, and this I learned how to create a new energy to the bacteria, and I learned a lot because I don't need a prototype of this how construct this project, and this project supports our global sustainability goals, especially clean energy, showing that small-scale innovations can turn waste into valuable energy, search and promote a circular economy. And that's all. Thank you for attention.